Tracing lineage in O-cluster mycobacteriophages through gene insertion and deletion patterns by Daria Nicki and Daniel Feeney. A little bit of introduction about O-cluster phages. O-cluster phages are very unique due to their unusual head shape. Cluster members share approximately 97% nucleotide similarity across their genome. We learned that that other 3% typically comes from patterns of insertion or deletion. The electron micrograph on the right shows an image of Blessica, which is an O-cluster mycobacteriophage that was isolated and annotated at the University of Colorado. A little bit of background about gene transfer. Horizontal gene transfer is the transmission of genetic material between genomes without inheritance from parent to offspring. An example of this is transduction, where viruses give their genetic material to bacteria. Vertical gene transfer, on the other hand, is genetic material transferred from parental genomes. And an example of this is how bacteria replicate and reproduce. And in our study, we specifically looked at horizontal gene transfer amongst mycobacteriophages in the O cluster. Potential horizontal gene transfer in Blessica. Blessica is at the top and Dylan is at the bottom. The green box on Dylan, which is genes 77 and 78, are highly conserved in the O cluster, but missing in Blessica. So this indicates a site of potential deletion in Blessica's genome. On the other hand, gene 82 in the red box is only found in half O-cluster phages, but is found in almost all L1 through L4 phages. So this is a site of potential insertion within Blessica's genome. After looking at that, we constructed a table comparing 8 of the 20 O-cluster phages. And we found that their similarity typically happens at the beginning of the genome, whereas their differences are later in the genome, which we know is typically characteristic of bacteriophages. So we constructed this table of differences. The genes are labeled at the top. And so we basically went through and said, is this gene here? Is it not? And so from this information, we constructed a phylogenetic tree showing the relative relationship between cluster O mycobacteriophages and a hypothetical common ancestor. So with that image, our common ancestor is at the bottom. Firecracker is closest to that hypothetical common ancestor with 3 differences, whereas CatDog is the furthest away from that hypothetical common ancestor with 14 differences. We see Blessica closer to the hypothetical common ancestor with 9 differences. And on average, it seems like phages ranged between 9 differences and 12 to 13 differences, with some outliers on either end. This is a famirator map of the hypothetical ancestral cluster O phage. This map was constructed based on a comparison of Blessica's genome to the 7 other cluster O mycobacteriophages. The red lines indicate positions in the genome where Blessica acquired genes by horizontal gene transfer from other mycobacteriophages, whereas the green boxes indicate families of genes that were not found in Blessica's genome but were present in other cluster O genomes. So this image overall represents a hypothetical ancestral O-cluster phage. Acknowledgements. We'd like to thank Dr. Guild and Dr. Philman for their support of this project, the MCDB 2161 class for their effort to annotate Blessica's genome, the MCDB department at CU Boulder, and HHMI and the C-Phages program.